Hippocampal and cortical neurons have been used extensively to study central nervous system neuronal polarization, axon dendrite outgrowth, and synapse formation and function. An advantage of culturing these neurons is that they readily polarize, forming distinctive axons and dendrites on a two-dimensional substrate at very low densities. This video demonstrates a technique to dissect, culture, and transfect embryonic mouse hippocampal and cortical neurons via nucleofection, which enables the expression of fluorescently tagged fusion proteins early in development, approximately 4 to 8 hours after plating. Expression of the fluorescently tagged proteins is maintained throughout the lifetime of the neuron for more than 2 months in culture. This enables the study of protein function during early developmental events such as axon outgrowth and later events such as synaptogenesis and synaptic plasticity. First, glial cells are isolated from cortical hemispheres of postnatal day 1 to 3 mouse pups and plated into flasks. After reaching 70 to 100% confluency, the glial cells are then replated on glass cover slips. Then, hippocampal or cortical neurons are isolated from E15.5 mice and transfected via nucleofection with plasmids encoding fluorescent fusion proteins such as red fluorescent protein. The transfected neurons are then plated in imaging chambers for short-term studies. For long-term cultures, glial cells on glass cover slips are inverted over hippocampal or cortical cultures to provide trophic support. Individual neurons can then be imaged to show the dynamic localization of fusion proteins throughout axons or dendrites of neurons.
Hi, I'm Erik Dent, an assistant professor of anatomy at the University of Wisconsin-Madison. Today, 3 of my undergraduate students, Chris Viesselmann, Jason Ballweg, and Derek Lumbard, are going to demonstrate a procedure for isolating